Hello, everyone. I'm a PhD student at the University of Liège, and in our research group, we mainly focus on the patients with disorders of consciousness. To study the complex concept of consciousness, we simplify this concept to two simple dimensions, wakefulness and awareness. Based on these two dimensions, the patients in disorders of consciousness can be categorized into different subgroups. For example, the patients in coma, they neither have wakefulness nor awareness. But when they somehow recover and they just have the ability to open and close their eyes, and also when they have some just reflexive movements and responses to the stimulation, we call them unresponsive wakefulness syndrome, or UWS, or vegetative state. When they have some non-reflexive behaviors, but still they cannot communicate with us, we call them minimally conscious. They are in the minimally conscious state, which itself decomposes into two subgroups of minimally conscious plus and minus, which MCS plus shows some language preservation behavior. And when they start to somehow communicate with us, or they can use some objects functionally, we call them emergence from minimally conscious, or EMCS. And there are some other groups that they are awake, they are aware, but because of some damages, they are paralyzed, they cannot respond to us. We call them locked-in syndrome, and usually we don't consider them as the disorders of consciousness. Different neuroimaging modalities have been used to study these patients. In this talk, I just want to speak a little about the high-density EEG and the method that we used. The method that we used to study these patients is graph signal processing. Simply saying, what is a graph signal? When you have a graph, a weighted graph, and you put some values on the nodes of the graph, you will have a graph signal. In the neuroscience, we usually are using time series instead of just some specific values. So we put time series on the nodes of the weighted graph. So at each time point, we will have a graph signal for analysis. But what is this graph? Some different kinds of graph you can define. For example, you can define a structural graph based on the tractography, the DTI signals. And then just study the functional structural relationship between, based on this signal. What we did, we just used the high-density EEG recordings, which is 256 channels of electrodes. And we just calculated we constructed the underlying graph based on the physical distance between the electrodes. So the electrodes that are near each other, they have the strongest weights of the graph. And then we just put each line is the recording of each electrode. We put the time series on the nodes of the graph, which is the electrodes, and then at each time point we made a graph signal, and then we calculate a parameter called total variation, which somehow says how smooth is your signal constrained to your underlying structural graph. We included UWS, MCS minus, MCS plus, EMCS, and healthy controls, and in each group, 10 patients, 10 subjects. And we studied just the patients that have somehow normal brain. They are not very severely brain damaged, because some of the patients are really brain damaged, and for example, some part of the brain doesn't exist. So if you want to do EEG analysis, it's a challenge. We just included some patients that have normal brain. The results was interesting because it shows that the total variation increases based on the level of consciousness. And in each group, for example, UWS patients show lower total variation, which means that the signal is more smooth on the structural graph that we have made. Also, we correlated this measure to a specific measure, which I wrote here, CRSR. It's a behavioral assessment, which is called Coma Recovery Scale, revised version, that they assess patients with some behavioral assessments, and then a score. And this score is a measure of the level of the consciousness. As it is more, the level of consciousness is more. And it shows again that this total variation increases with the level of consciousness. What does it mean? I will conclude my talk. The meaning is that based on this graph that we have made, we can conclude the communication and the signal are more segregated. So they, in the UWS patient, when the level of consciousness decreases, the communication between brain regions are mostly local. They are just the local regions are connected to each other. But can we say something about the long-range communication based on this graph that we have made? No, because the weight of the edge between long-distance nodes are almost zero, so they do not have any role in the total variation parameter. So the work that we are going to do now is to adapt this model for the long-range communication and to improve the underlying signal for further analysis. Thank you.